Joe Rogan has actually talked about this concept without phrasing it as PGPRs. He has had on multiple podcasters who have talked about these sort of nutrient rich soils in the rainforest. There are these legends. Hello, hello, hello. I am John Beckman, Professor John Beckman, Dr. John Beckman.
I am Worf.
Coming to you live from Southern Command with half my brain typing on my back.
I have much to teach you about women.
Oh, that's hot. Tea, Earl Grey, make it so. Okay. Ooh. All right, today I want to talk to you about a new publication from our lab that just came out entitled Fast Screening Libraries of Plant Growth Promoting Rhizobacteria, PGPRs, for Insecticidal Activity, or short title, Fast Screening PGPR Libraries for Insecticides. I am John Beckman, Professor John Beckman. You can follow me on X at Stampede Press. I would encourage you not to follow me. You can also check me out on Rumble and YouTube as Professor Beckman, Prof. Beckman. All right, let's get right into it. PGPR introduction. So PGPRs, plant growth promoting rhizobacteria, are bacteria that live in the soil that can help plant growth. There are many different mechanisms by which they can help plant growth. They can prevent pathogens, protect the plant, so sort of like stimulate the immunity system, kill pathogens in the soil that hurt the plants. Just like humans get sick, plants also get sick. So these bacteria can help keep the plants healthy. They can also promote growth. So they can promote root growth, shoot growth. They can produce plant hormones that help the plant grow faster, which is important for agriculture. So they can be a biostimulant, a bioprotectant, and a biofertilizer. They can also help the soils be more nutrient rich. So that's the gist of plant growth promoting rhizobacteria. I've got a lecture on plant growth promoting rhizobacteria, multiple lectures. I can post them if you want to learn more about that. Brief history of PGPRs. So I actually work at Auburn in the department of the original sort of one of the founders of PGPRs, Joseph Klepper. And he was the first person to coin the term plant growth promoting rhizobacteria. This dates back to an original paper, I think somewhere around 1980 again. That's in the original lectures if you want to learn about that. But what Dr. Klepper did in our department is he traveled all throughout the world and collected vast libraries of plant growth promoting rhizobacteria. So you can collect these bacteria that live in the soils. And most bacteria, well, actually not most bacteria, but many bacteria that you can grow, you can also freeze down in what's called glycerol stocks. So you can treat a bacteria with some glycerol, and then that allows you to cryopreserve it, cryofreeze it. So you can take these cultures of bacteria, put them in little tubes, and stick them in the freezer, and they can survive perpetually, forever in the freezer. You can take them out and you can reculture them. So what Dr. Klepper did is collect many, many, many different soil samples of bacteria from diverse soils, from diverse agricultural systems all throughout the world. And so Auburn has these gigantic libraries of plant growth promoting rhizobacteria. And when I originally started here, many of the smart people who I interacted with were getting involved with these libraries. So I kind of wanted to jump on board and see if there was anything I could do with these libraries that I would be interested in, that I could approach from my perspective. Of course, I'm an entomologist, so I study ways to kill bugs. So my original thought was, well, maybe we could screen these libraries, develop a screening methodology to figure out if we could develop a new insecticide from some of these bacteria. So in the same way that humans get sick, plants get sick, bugs, insects, especially pest insects, can also get sick. And so there you'd be able to find bacteria that might help the plant, but also kill the pests that are in an agricultural system. And you probably maybe you have not heard the term plant growth promoting rhizobacteria, PGPRs, but you probably have heard of this concept. Even Joe Rogan has actually talked about this concept without phrasing it as PGPRs. He has had on multiple podcasters who have talked about these sort of nutrient rich soils in the rainforest. There are these legends that the rainforest used to be like a cultivated fruit garden of many different trees that would sprout crops for people. And it was thought that the soils there was something special about the soils that would promote the growth of these fruitful trees, okay, that would foster these large civilizations. That's one sort of like interesting hypothesis of perhaps maybe 20,000 years ago, 10,000 years ago, maybe the rainforest had these societies that had these special soils that would grow crops and would allow them to amass large populations to construct civilizations. And it's thought that the knowledge of these super fruitful soils has been lost. And I think there might actually be some scientific merit to that idea. And it could all be encoded in the microbiota, the microbe communities that live in the soils. And that's essentially what plant growth promoting rhizobacteria are. They're cultures of whole ecosystems of microbes that live in these soils. And if you have the right mixtures of microbes, you can easily tell how that would be beneficial for a plant versus negative for a plant if you have the wrong microbial community. So this is actually like a well-established principle in the scientific community of people who study microbes. This is definitely like a real thing, that your microbial population will definitely impact the soil fertility and the cultures of different crops that you can grow in any particular soil. So that's a real thing. And I kind of wanted to start researching that, again, to see if there's anything that I could contribute and also to sort of like test the merits of this idea. Another thing that is sort of like an underlying hypothesis to this project that I am very interested in is this idea of microbe miners. So if I was ever to start a biotech company, if I was to start a biotech company right now, which I'm kind of like in the process, maybe like slowly, kind of thinking things through. Again, I like being a professor, but if I was to start a company, the company that I would start is I would just go around the world and collect microbes, because it's extremely easy to collect them. It's extremely easy to culture them. And every single microbe is different. You have no idea the vast diversity amount of microbes that live in the world. You can go to any different soil a mile apart and find vastly different microbes. Okay, so when people do like metagenomic sequencing of things that are in the soil, everywhere you find something different. And even if you find the same thing, what you have over there is different in genetic content from what they have over yonder. So even if you have, quote unquote, like the same species of microbe, actually the genes in those microbes are often different, what we call alleles, different variants. And there's often different plasmids, different genetic mixtures of genes that can completely change a phenotype. So for example, you've probably heard of E. coli. There's many different strains of E. coli. Only a few different E. coli's are pathogenic to humans. And actually in the lab, we study and work with safe E. coli strains. So every strain of bacteria is very, very different depending on the genotypic context of that bacteria. And so again, the idea of what I would do if I was to start a biotech company is just collect and amass vast collections of microbes. And you can organize these in sort of like 96-well plates. And then I would develop screening methodologies to figure out what they're good at. So essentially, it's like that you can envision that microbes are essentially like little Pokemons. And we have not even begun to explore the powers of what microbes can do. And so you could imagine you collect so many different microbes in these little Pokeballs that you can stick in the freezer. That's a real thing. And then you just develop screening methodologies to figure out what each one is good at. And through this system, you could develop very, very valuable microbes that could do all kinds of different things. We have no idea what they're capable of doing because nobody has really explored this to a full extent. So what this project was to test the merits of that idea, where we would take some of these libraries that had been amassed in the Auburn freezers, and we would test them for a specific phenotype that was useful for humanity, in this case, insecticidal activity. So many of the insects that we live with attack and kill and eat are herbivores of crops that we want to eat. So they destroy agricultural systems. And so we can screen these microbes to see if any of them will kill the pests that we need to kill and develop new insecticides through that methodology. So that was the premise of this paper, and it succeeded. So this is sort of a proof of concept that the microbe miners idea is real. And I do honestly think that this will be sort of like in the future, this will probably be some sort of gold rush, in that the thought is you could probably find a microbe that could do almost anything. The hardest part is just finding where is that microbe and how do you screen for the phenotype. That's the hardest part. And to, I guess, just further validate that this idea is very interesting, as soon as we published this paper, we actually did get contacted by a company who immediately licensed some of these strains. So this was actually the first project that I have ever done in the lab that actually generated like a tiny amount of money. So I actually feel like really proud of myself that I was able to maybe contribute something useful. But not to get your hopes up too much, let me also just add a layer of skepticism and cautious skepticism about this idea. There are some criticisms. The first is that if you find a microbe that's particularly useful, sometimes that phenotype, the thing that it does, is not super consistent. So sometimes you'll see effects of microbes, and sometimes those effects of the microbial communities can drift. And so one of the problems with PGPRs in the past has been that you might find something that's working in one particular context, and then you try to apply it to the field, and then all of a sudden, like, conditions change, the microbial population changes a little bit, and that phenotype kind of disappears. So there is the problem, sort of, that one of the problems towards commercialization is that these microbial communities seem to be a little bit inconsistent in the phenotypes that they produce. And another concern is that they're often location dependent. So many of these microbes have probably evolved to live in the niche or the environment that you found them in. And when you take them out and you sort of bring them into the lab, and then you maybe ship them to a different place in the world to sort of like use them in an agricultural context, then all of a sudden you're rapidly changing the environment that they evolved for. And so oftentimes those successful phenotypes disappear. So there's a lot of inconsistency with PGPRs. That's the skepticism. There's a lot of problems with figuring out how to apply them. But I really do believe in, and I like the idea, and I do think there's evidence to support the idea that there is a real thing called like super fertile soils. And part of what makes a soil super fertile is the microbial community. And so if we could understand the best microbial communities, we would be able to generate more fertile soils. I think that has sound logic to it. And that's what this paper is about. And again, I was skeptical of PGPRs at first. I did not want to, I would say I was hesitant because of the skepticism and the inconsistency. But then when I started reading the literature, there are some pretty convincing phenotypes that you can clearly observe in the literature. So here's a paper that I talked about in the past using and testing plant growth promoting Rhizobacteria in peanuts, which is a big crop in Alabama. So here are two examples of some of these phenotypes. So if you look at left versus right, these are germination trials with peanut seeds. And on the left, you're seeing plus PGPR, and on the right, you're seeing minus PGPR. So plus bacteria, no bacteria. And on the left, you can see that it's adding more nodules in the root. So these things that are popping out of the root, that is actually induced by the bacteria that you germinate the seed in. Compared to the negative control, these have much less nodules. In terms of like biomass, of peanut biomass, so here's another experiment where they're growing peanuts in the context of plus PGPRs. In this case, the plus is on the right. So here plus, and then on the left, there's a minus. You can just see in this part compared to the left, these peanuts are much more robust and there's a lot more biomass. And same here in this other replicate. And so there's lots of significant evidence suggesting that PGPRs can help plants. And so just by planting a seed with the right microbial cocktail, you can get a lot more yield in any particular crop that you're looking at. That's one example. Here's another example that's in our department. So this paper was published with the collaborator David Held. David works with turfgrass, and I also have some projects with David on studying turfgrass and how to make turfgrass grow more efficiently and protect it from insects. Here is, it's a little bit blurry, but here is a picture that David showed me right away when I first started here that got me interested in PGPRs because it's so visually compelling. On the left, this is turfgrass planted and treated without PGPR, minus PGPR. Here is turfgrass with plant growth promoting Rhizobacteria. Just look at the difference in the root systems of the turfgrass with bacteria versus turfgrass without the right bacteria. It's pretty shocking. And so this picture was one of the visual pictures to me that when I see sort of like a visual phenotype that is so robust, you don't even need statistics to tell whether it's working or not. You can just visually see that it's very robust. I always want to study things like that because if it's so robust that you can see it with your eyes, it means it's more likely to be real. So I really wanted to get into PGPRs. And when I first started PGPRs, I was a little bit more on the biotech side where I was thinking, well, maybe I would engineer the PGPRs. Let's figure out what causes their phenotypes. And just to be clear, this is probably where it will go in the future. Once we understand what causes these phenotypes, the genes in these microbes are entirely what causes their behavior. So you'll be able to take whatever gene you want, swap it out into a different bacteria, and induce that phenotype from a different bacterial system. So when I was originally getting into this, what I wanted to do was just engineer the microbes, figure out how to get them to do what you want them to do, and then genetically engineer them. I talked to Klepper about that, and Klepper advised me that is not going to be the fastest way to get something that comes to market. Because again, people are very cautious about genetically modified organisms, and rightly so. And so people are kind of uncomfortable with that. But what people are a little bit more comfortable with is finding naturally occurring bacteria that you might call these like healthy bacteria, good bacteria, probiotic bacteria that are natural out in the environment. And you can find these bacteria. And if you can just find one that gives you the phenotype, you could probably use that instead of genetically engineering. So again, that's what sort of propagated the screening methodology is, rather than engineering them, like let's not mess with their genetics. Let's just find a natural one that gives us exactly what we want. And then actually you could even take that bacteria and you could do what's called directed evolution. You could mutate it, and that is not actually inducing what's called genetic engineering. You could just mutate it and select for a better phenotype to increase that phenotype and the propensity of that bacteria to do what you want it to do through directed evolution. Okay. So again, that's kind of what we're doing here. We're trying to find natural microbes, good microbes, probiotic microbes that do what we want them to do. In this case, killing insects. Okay. So that's the context of like why I wanted to get into PGPRs, what this study was about, and the case setting up the rationale for this. Okay. So the way that we set up the study is we set up this tiered screening process. Okay, so what we call tier 1, this is sort of like the introductory tier 1 assay. So again, our goal was to find microbes that kill bugs. Okay, so the easiest bug to work with is Drosophila melanogaster. I've done a ton of work with Drosophila. It's the easiest test. So we're setting up a model to screen the bacteria against Drosophila, and we're going to check to see if they kill Drosophila. That's the tier 1 test. Okay, if the bacteria can kill some Drosophila, demonstrate some killing capacity for insects, then we funnel them into what we call tier 2. These are actual insect pests. Okay, so we tested them against aphids, Lygus, further tests of Drosophila melanogaster, fall armyworm, and a different instar fall armyworm. So essentially, if they can kill Drosophila, then we funnel them into these other tests against actual pest insects that are pests in the field. I'll talk about each of these insects later. And then a third tier was sort of like further tier screening of Drosophila with Drosophila suzukii, which is a blueberry pest. So I'll talk more about those insects down here. It's worth pointing out the actual library that we're working with. So we're working with the JM library. So this is a library collected by John McInroy, and it's an endophyte library. So endophytes are particular bacteria that can colonize inside of plants. So they will go inside of the plant. And so the reason we wanted to test endophytes is because the hypothesis is that if the bacteria can get inside the plant, then when an insect eats the plant, it will uptake some of these bacteria. So you're essentially creating a situation where these bacteria are more likely to be eaten by the bug that's eating the plant if they're endophytes. So that's an important distinction here of the library that we chose strategically to screen against. Okay, so let's show you a schematic of the experimental system that we're working with, how we're setting up these initial experiments. Okay. So this is what you would call like a library that I was talking about. You have these glycerol tubes, you have these tubes, and you culture the bacteria, and you just add a little bit of glycerol, and then you can set up these boxes of tubes, and in each of these ones is a different microbial Pokemon, okay, that you can pull out, and you can pull these out, and you can streak them out on a culture container. So we grow these, or in this case, you're growing them in liquid cultures. This is growth in liquid, I think this is TSB, tryptic soy broth. You see, we're growing these cultures in TSB for 72 hours. And the premise of this was that we don't quite know how the bacteria might kill the bugs. They might infect and be pathogens, or they might produce some chemical that through accumulation might kill the bug. So we wanted to grow these for sort of like a long period of time to get them to full stationary phase and allow them to produce whatever metabolites or chemicals they might produce. Okay, so we grew these for 72 hours, and then we would take an aliquot of these growth mixtures, and we would plate it on apple juice plates. Okay, apple juice plates are what Drosophila melanogaster likes to lay eggs on. So to get to Drosophila to lay eggs, it's very easy. You make what's called yeast paste. So Drosophila like to lay their eggs in rotting fruit. And so yeast will be growing in rotting fruit. So they have very strong odor sense for yeasts. Okay. And because it's on an apple juice plate, they think that it's sort of like a rotting apple with yeast on it. So you put this yeast paste on the apple juice plate, and then you put flies in the container, about 200 flies, and then they'll just start laying eggs, dumping eggs. Okay. So you can collect a ton of eggs. Then once we collect a ton of eggs, okay, we transfer 10 of these eggs onto the apple juice plate that's been treated with the microbial cocktail. And then we come back 4 days later and we count the survivorship. So we count how many dead larvae are there or how many living larvae are there. So it's a very simple assay. And so every single one of these plates gives you a number out of 10, how many survived the treatment. Okay. So it's a very simple test for just insect killing. Now, some criticisms of this, if we're going to really go into like lots of the details, which that's what this discussion is about, since it's the paper discussion. Some criticisms of this assay are that it might not be a good idea to test with Drosophila because insects are very different, and pest insects are not all dipter and flies, okay? And many of them are not related to Drosophila in any way whatsoever. They're very, very distinct and very diverse. And so if you start the screen by testing on Drosophila initially, you're going to lose a lot of potentially good microbes that don't kill Drosophila but might have activity against other insects. That is a legitimate criticism of the experiment. On the other hand, as a scientist, you have to figure out how to actually do an experiment that's feasible, that will actually work. And so Drosophila is, in any case, a good model because it will not get us all the microbes, but it will get us some of the microbes that aren't able to kill Drosophila. So that's one of sort of the main criticisms of this, is that there will be false negatives as an inherent result of this process. But as long as you understand that and you understand the caveat, then like there's no problem with that. Okay, so here is the first data for the first 500, 600 strains that we screened, okay? So we screened a ton of these strains. And an interesting thing is that this is only a tiny fraction of the library. The library is about 2,000 microbes large, and we screened about a fourth of the library, okay? So if you look at this graph, on the y-axis is the number 10 at the top. That a 10 up here would be that all the larvae survived, okay? And a 0 would be that they were all killed. And each of these dots is a replicate of 3 experiments. So 30 total Drosophila were tested to generate one of these dots. And each of these dots is a different microbe that was tested against the 30. So N is considered one experiment of 10 larvae, and each microbe got 3 of those experiments for the initial tier 1 test. So again, one criticism of that is that the N is not super high. But again, if you're screening 600 microbes, as a scientist, you're always trying to figure out, okay, well, what's actually functionally possible to do the test? And so we started with an initial 3N to see if any of these showed any patterns of killing, okay? And so then we color-coded the dots. So blue and purple are negative controls. One of these is a negative no bacteria, and one of these is a top 10 E. coli bacteria that should have no killing, okay? So essentially, like, the things that should not kill are right up here. And then anything below that is probably doing some killing activity. And so you have to decide where you're going to sort of set the threshold. So we decided to sort of set this arbitrary threshold that of bacteria that we were interested in must kill below this 6 number. So that means they must kill 4 or more Drosophila in order for us to be interested in them. And that's all these red dots, okay? So these red dots are killing greater than 4 larvae killed, okay? So that's sort of like the first round again. And some of these now, you have to understand, some of these in any screening process, there's false negatives and there's false positives. I've done lots of screens, and this is always an inherent problem of screens. So you need a strategy to reduce the false positives. So in order to get rid of the false positives, the ones that killed 4 or more larvae, we then did a second round, a tier 2 screen. So that's how this moves to this right here. Of the ones that killed 4 or more, we screen them again. And in this case, we increase the N to 10. So in this case, in that second tiered Drosophila melanogaster strain, we're now testing them against 10 plates, 100 total larvae. So it's a robust system to rule out false positives. And you can see there are many false positives. So again, here is the tier 2 data in Drosophila melanogaster. Same graph, same type of stuff. And you're seeing that some of these essentially stop killing. So the phenotype is inconsistent. Those are the black dots. The red dots are ones where the phenotype was consistent and it kept repeating. Okay. There is a lot of standard deviation. So we are seeing a lot of variation in some of these bacteria, although some of the bacteria are pretty tight, like this one, which is killing essentially all the bugs. So we're seeing some variable efficacy, but the screen in general, this tells us that the screen is working and we are finding microbes that can kill bugs, Drosophila melanogaster. Then of these ones that persisted and killed Drosophila up to the 10N replicate, those we funneled against the first actual pest, Drosophila suzukii. So Drosophila suzukii is a blueberry pest, and Drosophila suzukii is a unique bug. It's a unique insect because it is a fruit fly that will lay its eggs on ripe fruit, not rotting fruit. So Drosophila suzukii is agriculturally very important because it will destroy blueberry crops while they're ripe, not when they're rotten. Okay, so this is a huge problem in America. We have this all over the United States, and it causes billions of dollars in damage. And it's not just the blueberry industry, it's essentially like the berry industry. All berries are targeted by Drosophila suzukii. It's a huge problem. And so again, here is part C of this first sort of big data figure. You can see that some of these are killing Drosophila suzukii. So now we have natural microbes that we could apply to blueberries that kill Drosophila suzukii. So these are now going to get funneled, not in this paper. Now, sort of this paper was just to find the interesting microbes. But next, now in the future, where we'll go with this project next, is now to start testing these strains in greenhouse trials of blueberries. See if we can protect them from the Drosophila suzukii using these strains. Okay, and then of the tier 1 killers, we then started funneling these into tests with different pests. So this is really a very interesting applied microbiological and entomological paper. There's a lot of entomology in this paper, a lot of different bug systems that we tested. So the next thing we tested on is fall armyworm. Fall armyworm is a huge worldwide pest of corn and turfgrass and a lot of other things, but we're primarily interested in it for corn and turfgrass. Fall armyworm is so devastating that the final instar larvae can essentially like wipe out whole golf courses. Okay, it will destroy huge patches of grass. And so you want to try to find something that you can treat in the golf courses to protect them or provide some protection of the turf grass against the fall armyworm. That's the context for setting up this research study. And it's the larval stage that is the problem. Now, fall armyworm was actually very difficult for us to work with. Again, you might think that these experiments are easy because it's just taking microbes and putting them sort of like on media and then like adding a bug. It's a lot more difficult than that because you have to set up systems where you can grow the bugs. And growing and dealing with different bugs, every bug is a different animal and every bug is like a different kind of pet. And so you have to figure out ways to rear these and culture these and keep them happy and keep them safe while you're doing the experiments. And so each insect is difficult in its own way to work with, and fall armyworm was particularly difficult. One of the things that makes fall armyworm difficult to work with is that it's cannibalistic. So if you do experiments where you put multiple caterpillars together, they'll eat each other. Okay, so you destroy and sort of like conflate your data by mixing the fall armyworms together. So then you have to set up things individually, or you have to figure out strategies of how you can prevent them from cannibalizing each other. Okay, so it's difficult to work with. The other reason that we were having difficulty is we did not have a good positive control killer for fall armyworm. So there is a strain of bacteria called Bacillus thuringiensis, which is well known supposedly to be a strong killer of fall armyworm. And it is, it definitely is. Like there's the literature on this is very strong. And so what we did is we tried to find a strain. We went to the sort of agricultural supply store and got strains of BT that farmers actually use for fall armyworm. And we tried to develop those as a negative control, but we did not see much efficacy. So here the blue is BT. And what we're measuring here is first instar fall armyworm. So this is a kill test of first instar fall armyworm. So again, these dots here are representative of means where we're treating and we're testing 10 different fall armyworms. And BT is killing a little bit, but the standard deviation is within comparison to what's supposed to be the negative controls. So in this case, the blank TSB and the food only, we're showing lots of killing. So essentially what this is telling you is that fall armyworms in early instars are very difficult to keep alive. They're essentially like dying for whatever reason whatsoever. And this is kind of sort of like very stereotypical of insects. Insects are what are called spray and pray. They produce hundreds and hundreds of offspring. Most of those just die. And there's only a few small subset that actually survive to adult stage. So essentially that's one other reason why the fall armyworm were difficult to work with is they seem to be sort of like dying randomly. So harder to test, harder to tell what's going on here. Okay. So this first instar graph was essentially like, well, maybe this is not the right way to do the experiment. So then we started testing against third instars. Okay, so these are a little bit older, a little bit more robust caterpillars. Okay, and in this case, we do the experiment and we actually do see some strains that are killing third instars, and their standard deviation is different such that they are actually statistically significant from the non-killers. But in this case, the BT control, the blue control, is not actually killing them. So the BT strain that's supposed to be killing them, that the farmers are using to kill the fall armyworm, is not doing shit. It's not actually killing them, and so there's lots of weird things going on with the fall armyworm data here. But at least we did get some strains that are statistically significant from the non-killers, and they are producing a killing effect in the experiment with the third instar fall armyworm larvae. So the fall armyworm data is kind of like, it's kind of like a wash. Like maybe it's working, maybe it's not working. we don't really know. We need further testing. So what these are now getting funneled to is we're actually now doing greenhouse and field trials of these strains on actual turf grass in the field and in the greenhouse. So these have moved to now sort of like a secondary publication that will come after this. And actually one of these in the initial field trials that we're testing did seem to show equivalent activity as a standard insecticide against fall armyworm. So it's looking like this was successful in at least generating some microbes that kill fall armyworm. But there are lots of problems with this data in particular. Okay, then we tested against aphids. So aphids are a pest of cotton. Aphids spread all kinds of diseases, viruses. So they transmit viruses, they're vectors. And they also just hurt the cotton itself by sort of like sucking out the juices. So again, working with aphids is difficult in its own way. You have to grow cotton to culture the aphids on, and then you have to grow the aphids, and then you have to transfer the aphids to the particular experiment. So the way that we set up this aphid experiment is we had a tube, and you could take individual Apterus adults. These are wingless adults. You can put them in the tube, and then you can put what's called parafilm. Scientists will know what parafilm is. It's essentially like a thin membrane that lots of scientists work with. So you can put a thin membrane at the top, and then you can put a test solution. So this is the liquid that's inoculated with the bacteria in between two sandwich layers of parafilm. So you're essentially creating like a little sandwich for the aphid. And aphids have a stylus, okay, that they'll stick through the membrane and they'll start sucking the juices of the test solution. So you're essentially just, this is a feeding test. We're just feeding them the different bacteria of the ones that were able to kill Drosophila. And we're seeing if any of those can kill aphids in a feed test. Okay, so each of these tests with these bacteria and these aphids is essentially like a Bernoulli test. It's a yes, no, dead, or not. And the number of N's we got for each strain is essentially the number of boxes. White is a death, pink is a living. So here's our negative control, top 10. So the negative control in this case is actually working. The TSB is also a negative control. That's working. Okay, so essentially it's not killing very many at all in the case of the negative control. So this is just, again, E. coli that doesn't do anything. And in this case, we actually did see some positive control action. So this is the same BT strain. In that case, it is actually able to kill some aphids, which is interesting because the BT strain that we're working with is not supposed to kill aphids. So we are seeing some weird like flukes with the BT stuff. So I guess I would say to farmers, when you're picking stuff up, it might not actually do what it's saying it does. That's one thing that we're observing directly in the actual experiments. The BT that's marketed as a fall armyworm killer is not actually killing fall armyworm, and it is killing aphids, which is marketed as not being able to kill. So that's kind of bizarre, but that's what the data show. Okay, and then these other strains that are starred, these are strains that are showing significant killing activity. Okay, so then these strains now, the ones that are starred, these are now funneled into greenhouse tests in cotton. So we're now testing these in the greenhouse on actual plants in cotton to see if we can bring these to market. And these are the ones that the companies were quite interested in, the aphid killers. All right, and then the final test was Lygus. Lygus is also a plant of a pest of cotton. It's one of the most prodigious pests in cotton here. Lygus is actually more important than the aphid in Alabama. And so in this case, the setup is, again, you have to set up all these different experiments a little bit different. In this case, you put the Lygus in a container, you have this what's called a foam, a floral foam. So it's just sort of like a green foam that the Lygus sucks juices out of in these cups. And then we add the inoculated media to the floral foam and let the Lygus bug drink the bacteria. And in this case, we saw a ton of killing. So these are the strains that were tested, and we got a ton of killing. And so now, again, finally, these are now in greenhouse testing. And then in conclusion, final figure of the paper, we sort of have just compiled all the information. So we have all the strains that we're detecting killing activity for of individual pests listed and their efficiency of killing. So some of these are 100% killers of certain pests. Okay, we've marked those, and we did some of the library strains were already sequenced a little bit with some older methodologies, PCR-based methodologies, and some of these ones that we're interested in now, we're funneling to genomics. So we're sequencing their genomes. We've sequenced a few of these genomes, and then we're going to do genomic comparisons to see if we can figure out what are the actual genes that are killing the bugs. Once we find the actual genes that are doing the production of the material that's killing the bugs, then those can actually be turned into chemical insecticides. So it's again, it's another sort of like mining strategy for finding new insecticides. but that's future work. That's the next steps of this. And it's also worth pointing out that some of these, I'm looking at this now, triple, that's Rich. Blame Rich for that. Anyway, so some of these bacteria will kill quadruple species. So they'll kill 4 different species of insect. Some of these kill triple species, and some of these kill double species. So some of the interesting things is that in some cases we're seeing insect-specific mortality, and in other cases we're seeing insect-broad mortality. So you do have to be careful about some of these things, right? Because you don't want to put stuff out that's going to be killing honeybees. So that will have to be dealt with in the greenhouse tests and before you do significant field trials. You have to test for what's called off-target effects. You don't want to be spraying into your field things that kill all kinds of different bugs. That could be a bad case scenario. And one of these, so Serratia is actually one of the strains of bacteria that the Navy sprayed all over the United States and actually killed a person. So again, some of these you have to be careful with. That's where the greenhouse trials and slow field trials become very important to figure out what's safe. All right, so that's kind of the gist of this. This work was done in collaboration with Dr. Martin, Dr. Held, Dr. Lawrence. This was Rich's big PhD project. And then we also had help from Jasmine, Janaya, and Bella on this. And this was funded by Alabama Cotton Commission and the United States Golf Association, Davis. And then my research salary is in part helped by USDA and NSF Career Award. Have a good day.
I am Worf, son of Mog. I have much to teach you about women. Notice how the Klingon lures a mate. Klingons do not pursue relationships. They conquer that which they desire. Today, I am a warrior. I must show you my heart. I travel the river of blood. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am Worf, son of Mog. I have much to teach you about women. Notice how the Klingon lures a mate. Klingons do not pursue relationships. They conquer that which they desire. Today, I am a warrior. I must show you my heart. I travel the river of blood. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. Worf, son of Mog. I have much to teach you about women. That is how the Klingon lures a mate. Klingons do not pursue relationships. They conquer that which they desire. Today, I'm a warrior. I must show you my heart. I travel the river of blood. I am a Klingon. Doubt it. A demonstration can